Okay, good morning and welcome to what is our first Ask the Author webinar, which is the beginning of a series that we're looking to be here every month. So welcome, make yourselves comfortable. Please come on in. I can see people arriving. Just to say that although we are on Teams, we are in a format that is for webinars. So you will find that your microphone and your camera don't work. So please don't worry about that or start bashing your computer trying to make it work. That's a centrally set up thing. So please welcome. My name's Matt Reed. I'm going to be your host this morning. We have an hour of discussion and talk. You'll see that you have an opportunity of two buttons. There is a chat button, and I'd really like it and welcome you to introduce yourself in the chat, say where you are, say what you're interested in and what you're working on. And then you'll see a Q&A button, which is questions for our speakers this morning. So please use the Q&A that you would like questions addressed to the speakers, either now or as they are speaking. And then there's also the chat. So please use that to introduce yourself. We have our speakers with us, and we also have some colleagues who are busy making all of this work. So you'll see Jane, Charlotte, and Honor, who are collecting your questions, who are making all of this work in the background. So please don't worry about overwhelming them. They will really enjoy all of your questions in your chat. So please jump into that. And then also in the chat, we will be posting little extras, little things that you will find helpful for the morning, and then things that you can look up later and enjoy and will add to the whole experience. So it looks like we have people all arrived. I'm hoping that that's the case. That's looking good. So we're going to move to our first presenter. We have two people speaking this morning. Well, Vera's going to start and she'll be talking for 10-15 minutes, and then we will move to Dennis, who will be talking for about half an hour, and then we'll have time for questions. So I'd just like to hand over the floor to Vera, and we'll look forward to Vera joining us.
Thank you very much, Matt, for the introduction, and welcome everyone also here joining us today. So I will be brief. I will share now my slides. I hope you can see them now. And I will be brief. I will introduce mostly the project. My name is Vera Silva. I'm a researcher at Wageningen University, and I am the project manager of Sprint and in the coordination team of Sprint. So I'm very happy to be here today to launch also this series. So short introduction on the project. So this is a European funded project, and tackles the big topic of integrated health approaches and alternatives to pesticide use. So rather big challenge. It's a 5 years project, so it started on the 1st of September 2020, and we are entering now the last year of the project, which will be ending 31st of August 2025. I put here in the slides a link to an explainer video. I believe you could also find it in the chat, and it's a 2-minute video that you can later on explore, that summarizes a bit of the project approach. And of course, for detailed information, you can also look into our Sprint website. So in a nutshell, so the main objective of Sprint is actually twofold. First part relates to the assessment of the risks and impacts of plant protection products, so the pesticides that are actually used under an agricultural context. And we look into the risk of impacts of these PPPs on ecosystem, plant, animal, and human health. At the same time, we also use the knowledge and the data that we generated and that we gathered to work towards more sustainable food protection. It's a big challenge. We don't do it alone at all. We have a big team across Europe, but also a partner in Argentina. And we have a team of different experts, multidisciplinary team, and you can see here in the map the location of our partners, and in the right the logos of all the partner institutes involved in the project. Here in this slide, some highlights about Sprint. So I think the starting point is that we use a holistic One Health assessments. So, and this covers indeed the coverage of environmental and also biological distribution of pesticides. We also look into different health endpoints. We have also generated unique data sets, both in the field and in the lab. So this series, you can also get familiar with some of these data sets and their findings. We have also developed new approaches focused on the testing of mixtures of pesticide residues. And we have also a key output of the project that is the Global Health Risk Toolbox. I come back to this in a second. That makes integration of the findings. Last but not least, the whole project has a big foundation on the transition and on the engagement of stakeholders. And this foundation indeed lies on the fact that we can only move towards more sustainable production if indeed we are engaged with all different actors of the food chain. So we have this kind of slogan on the all aboard of the Sprint transition train. So in this series, we'll get presentations from the different type of peer review articles. So up to now, we have 34. All of them can be accessed through our Sprint website. I put here the direct link to them. And these cover, from instance, from the field study protocol that we did to findings of pesticides in a specific matrix, like what is today the case on soil. But we also have some publications where we do make overviews, or where we present analytical developments and so on. And in this slide, in orange, I also highlighted some of the data that you may find in these publications that goes from numbers and levels of pesticide residues, but also where we do some applications of this information, of this monitoring data, for instance, with an example here of a development of a pesticide prioritization indicator that can be used for regulators and policymakers. Here in this slide, you have an overview of the field campaign. So Sprint started in 2020, and we started our field campaign 2021, just at the same time as of COVID pandemic. So it was challenging times, and but we succeeded. I think here we have to say huge thanks to all our case study site teams and all the participants that made it possible. But here you can see in the left side of the slide a map of our 11 case study sites. So each one of these case study sites included conventional and organic fields. And altogether, they cover the main crop types in Europe, and rather often they also had livestock. We had Argentina as soya crop example, also from the import to Europe from feed. In each one of these case study sites, we did a rather comprehensive data and sampling campaign that you can see on the right side, and where we collected samples from human participants, from ecosystem, livestock, and even companion animals. So for instance, in the human part, we had collected samples from 3 population, so the farmers from both conventional and organic fields. We have also collected samples from the neighbors that live nearby these agricultural fields, and even consumers that live at greater distance rather often in cities. And from each one of these participants, we collected urine, feces, blood. We also asked them to wear a silicone wristband so that we could also infer on dermal contact. We have also performed another type of nasal swabs, not for COVID tests, but to assess microbiome. And we also asked them to provide us samples from the food, so we could also look into dietary exposure. So this for humans. For ecosystem, for instance, we have soils, sediments, waters. We had indoor dust. We also analyzed pesticides in earthworms. We also look into the diversity, for instance, of flying and ground-dwelling insects, and so on. Here some pictures of our field campaign, and also some numbers on what it entails. So you can see that we had around 1,800 environmental samples, 1,400 livestock samples, and around 4,000 human samples from 657 participants altogether. So not all of these samples were used for pesticide analysis. As I mentioned, we also look, for instance, to diversity and microbiome, but most of them were. And in each one of them, we tested, we looked to 209 pesticide residues. That was the target. It was not always possible in all the matrices, but that was what we targeted. Now I move to the lab work that we conduct, and here you can have a glimpse of the ecotoxicological, so where we look into effects on terrestrial and aquatic ecosystem, but also into the toxicological aspect. So we also do tests to infer on the risk of the mixtures that we found in the field towards animal and human health. These tests are also being finalized at the moment, so hopefully later on in this webinar series you can also get access to some of this. The key output, as I mentioned from the project, is this Global Health Risk Toolbox that is, well, a box of different tools, which can be also applicable for different end users. That goes indeed from regulators, policymakers, but also for industry as well. So we also have an animation, a Prezi on this toolbox that you can dig later on to understand also how this toolbox had been put together and the type of potential of the different tools. Last but not least, about the transition part, we work very closely with the stakeholders from the beginning until the very end of the project. And we work with them on two main areas, on awareness raising, of course, on the risks and impacts. We ask them also to provide us samples, we report back the findings. So that is one of the areas. The other one is to work with them to understand and design transition pathways. So understand what are the barriers and the opportunities towards other type, more sustainable production with less reliance on synthetic pesticides. And we do this also considering each context of the different case study sites and the different dimensions of the transition that it entails. That goes, of course, from knowledge, cognitive dimension, but also economic and policy related. So that altogether, we are also working towards policy recommendations and the research agenda to tackle some of the still knowledge gaps around pesticides and sustainable food production. I think that's it from my side. I guess this is Sprint in a nutshell. If there are any questions, I'll be happy to answer it later today or in the chat. Thank you.
Thank you very much, Vera. Always great to hear about Sprint. And that is Sprint in a nutshell, a very large nut, perhaps, because there's quite a lot to understand. So Charlotte has peppered the chat with how you can find out more about Sprint if Sprint is new to you, and also how you can follow Sprint and interact with it, because it's a lot of exciting and I think impactful things happening. So thank you very much, Vera. You'll be back later to answer questions, and I'm sure people will be able to pose questions that might occur to them later. Now, friends, before we move to Dennis's presentation, I just have a quick apology. I should have mentioned to you earlier that we are recording the webinar. So I don't know how impactful that is on your privacy if you are somewhere you shouldn't be or with somebody you shouldn't be with, and that somehow that is apparent on the webinar, then please take some action. But otherwise, just for your note that we are recording it so we can play it back to people later. And obviously, we're very attentive to your questions and interested in them. So we will be following them up. So part of why we're recording this is to do that. So now I'm going to hand over to Dennis, and you're all good to go, Dennis, by the looks of things. So over to you.
Can you see my slides?
Absolutely.
Perfect. So I'm Dennis Knut. I'm doing my PhD in the Sprint project under the supervision of Vera in the soil physics and land management group at Wageningen University. I'll start this presentation as the target audience was not necessarily academic or from the field of pesticide analytics or pesticide use, with a short introduction to pesticides, like history-wise, and then the context of the study, how the study was performed, and then some results and conclusions. So what are pesticides? In general, pesticides are substances that are used to kill, repel, or control a certain form of pests. So fungicides for fungi, herbicides for plants, and insecticides for insects. There are obviously also other kinds of pesticides that are bactericides, rodenticides, repellents, nematicides against nematodes. But normally we use mainly the three classifications of fungicides, herbicides, and insecticides. Pesticide residues, as we always call them, are either the active substance in a pesticide, or co-formulates or degradation products of pesticides, of the active substances. A look into the history shows the first recorded pesticide uses was about 4,500 years ago. That was sulfur as an insecticide in Egypt, I think. Synthetic pesticides emerged in the 1930s, 1940s, with, for example, DDT or 2,4-D. DDT became widely used for everything, like from pets to humans to crops. Everything was that needed to get rid of insects was treated with DDT. In the 1970s, 80s, glyphosate was introduced to the market and became the most sold herbicide in the world. With this emergence of synthetic pesticides and other agricultural techniques, fertilizers, and improvements in technology, the Green Revolution came to place. And it's not this green revolution, but it's the industrialization of farming. And this was needed and had definitely good sides. So the crop yields increased, for example, for barley from 1.3 tons per hectare to 3 tons per hectare between 1960 and 2021. for wheat from 1.1 ton per hectare to 3.5 tons per hectare, and for potatoes from 12.2 tons per hectare to about 22 tons per hectare. This increase in production was also necessary as the world population grew from about 3 billion people in 1960 to about 8 billion people now. And a big part in the Green Revolution are pesticides. The European Crop Protection Association says right now still 40% of the global crop yields are lost to pests and disease, and without the use of pesticides, it would be twice the amount. But pesticides have also shown a negative side. In 1962, Rachel Carson published a book, Silent Spring, that put attention to accumulation of pesticides in the environment. So elm leaves were sprayed with DDT. The leaves fell and were digested by earthworms, which were eaten by the robin. And this the DDT was toxic to the robin, and so robin populations decreased. Similar, the bald eagle, when they ingested food that was DDT contaminated, their calcium pathways were disrupted, and the eggs they laid were not able anymore to sustain the weight of a breeding bird, and so the eggs broke, and also their populations were going back. So relatively new, October this year was an article in the Netherlands and Dutch news NL that pesticides that are still used in the Netherlands are killing birds and bees. This is a result of a meta-analysis by Molenaar et al. From Wageningen University, which take a look at neonicotinoids like imidacloprid, acetamiprid, and thiacloprid. So what happens when pesticides are used? This slide is a bit out of place, but I think it's very important to just look at this. We apply pesticides, there's spray drift. So pesticides are going off target into the environment. They can be washed off of plants, and several degradation pathways or pathways out of the soil ecosystem, agricultural ecosystem, like leaching into the groundwater or volatilization, photodecomposition, chemical degradation happen. The state in the EU is that about 450 different active substances are approved, and we use about 470,000 tons per hectare, tons a year, and we aim to decrease pesticide use as well as pesticide risk by 50% by 2030. The governing body here is the European Food and Safety Authority. How do we assess pesticides? We have field campaigns, screening. There I'll have some examples later. And these screenings are often just like not the 450 different active substances under investigation, just a small part of it. Often just a small agricultural area or in a single country. The studies are varying in sampling time and sampling strategies, so there are problems with comparability of these screening campaigns. One of the largest screenings in Europe, or the largest, is the Lucas project, where they, for pesticides, used 3,473 samples, analyzed 118 substances, and found that 26% of these samples were pesticide-free, and 57 samples had multiple pesticide residues. 11% had over 10 different pesticide residues. Here is to say that in Lucas, not just agricultural areas have been sampled, but also other natural areas, other soils. Another also Lucas samples, but from another year, was the screening from Silva et al. They used 317 samples and analyzed 76 different pesticides and found multiple residues in 58% of the samples, and 17% of the samples were pesticide-free. These were just samples from agricultural areas. So these screening campaigns are very labor intensive in the lab and in sampling. So alternatively to screening, we also do modeling. We predict the pesticide concentration in the environment, for example, with the ESCAPE tool, where we have input parameters like the pesticide application rates, then degradation kinetics and degradation times, as well as numbers of application, the interval of the application, how much of the pesticide is intercepted by a crop. And in Sprint, in my paper, basically, or in the study I wrote about, we sampled 201 fields from European agricultural areas and analyzed them for 192 different pesticide residues. So the target, as Vera told earlier, was 209 different pesticide residues, but we were not able to analyze 17 of those reliably. Then there were questionnaires about the pesticide applications from the farmers. There were extensive questionnaires, and one question or one part of it were about application rates and application intervals of their plant protection parameters. And with this information, we also calculated the predicted environmental concentrations of the pesticides and compared them later with the measured concentrations. What we found of the 192 different pesticides we analyzed, we found 100 different, 96 different in conventional fields, and 48 different in organic fields. We found significantly different numbers of pesticides in organic and conventional fields in 7 out of our 10 different case study sites. There were no significant difference in Croatia, Italy, and Slovenia. In Switzerland, Czech Republic, Denmark, Spain, France, the Netherlands, and Portugal, we had significantly lower numbers of pesticides in the agricultural fields, in the organic agricultural fields, than in the conventional ones. Similar found Geisen et al. In a study that analyzed 151 different pesticides in 340 different samples, where they found more than 6 different pesticide residues in 92% of their samples. We compared also from the questionnaires which pesticides were applied and detected and which were not applied but detected. So 51.5% of the pesticides that were applied, we could also detect. So that means 48.5% that were applied we could not detect. This is possible that they were already degraded when we sampled. But more interesting, or also interesting, is with what we detected but what wasn't applied. So we detected 642 times a pesticide that was not applied in the current growing season, which means this can be through transport from somewhere else or just a legacy from former applications in years before. So, total pesticide concentrations were significantly different only in 5 of our 10 case study sites in Spain, France, Croatia, the Netherlands, and Portugal. When we look at the comparison between prediction and measurement, you see the line in this graph is 1. So if prediction and measurement would be the same, the points would be on the line. Everything below that line, the measurement was higher than the prediction, and everything above the line, the prediction was higher than what we measured. In general, we just see that we have a high variance between prediction and measurement. This is basically the same graph, just divided in the pesticides, the active substances, compared to literature or other studies, for example. So we see that there's high variance between over prediction or under prediction. The measurement and prediction are just off. And other studies found similar results. So Rido and Wächter calculated new degradation times for their pesticides from what they saw in the field, and saw that their degradation times are higher than what they find in the literature, what would be used for the predicted environmental concentrations. On the other hand, Fougere et al. Found mainly under predictions. So in their study, the measured concentrations are mostly not exceeding the predicted concentrations. Why are these pesticides a problem? So a study from this year, I think, actually showed the effect size of different environmental global stressors on soil biodiversity. And number 1 stressor is pollution. And they divide then pollution in pesticides and metals. And metals are worse for biodiversity than pesticides, but pesticides are a contributor to declining soil biodiversity. Here is to be stated that some metals are also used as pesticides, and I don't think they divided in their study between metals that were used as pesticides and just metals. Some studies show that there is a risk of pesticides to soil organisms, like from Jakob Hoffmann's lab. They show that pesticide residues in soils pose a risk at 35% of their sampled size or their analyzed size, and that there's a lack of availability in toxicity data of pesticides and transformation products. Celine Pelosi's lab found that pesticides pose a risk to soil invertebrates. The same group also asked if pesticides in soils are a risk for earthworms. Why is biodiversity, earthworms, etc. important? They're responsible for bioturbation, nutrient availability, that's macrofauna like earthworms and mites. For the nutrient cycles, so nutrient availability for plants, for example, for soil fertility, carbon storage, and aggregate stabilization. This is done by microorganisms, for example, bacteria and fungi. We figure out how or if active substances or pesticides are dangerous for soil organisms, for example, in ecotoxicological tests. So pesticides are applied to soil and to see if organisms have a negative effect, are negatively affected by this. This is mostly done just on a single species of organism. In a lot of cases, especially for approval of pesticides, just with one pesticide, and then also just with the active substance, not with the whole formulation. Yeah, and we still try to reduce the risk of pesticides by 50% by 2030. So our conclusion from the study was, as under field condition, pesticides mainly occur in mixtures. Also for approval and the risk assessment, we should use mixtures of pesticides. Organic management shows in most cases significantly lower numbers and concentrations of pesticides. So change to organic management can be still a tool to achieve pesticide risk reduction to 2030, and also the reduction of pesticide use and less use of persistent pesticides can reduce the pesticide risk till 2030. So that's from my side, more 20 minutes than half an hour. But
Dennis,
if you have questions,
that's great. Thank you very much indeed. Thank you, and thank you for the round of applause there. It's an opportunity to ask questions, isn't it? So there is a lot there, I think, for people to discuss and talk about. So that's really exciting. Vera's going to join us again. So hello, Vera. Nice to see you back on screen. Just to say that in the chat, you will find links to Dennis's paper. You'll also find links that hopefully we get to work on summaries and other materials that sort of help provide some context and background for what we've been talking about today. So we have some questions in the chat, and also when people registered, they also left us with some questions. So let's get into those questions. So let's start with maybe straightforward one, but why not start with a straightforward one? So either for Dennis or for Vera or for both. Question: given that pesticides are designed to kill pests but aren't species specific, can we assume they harm all soil-dwelling organisms, thus negatively affecting soil health and nutrient transfer to crops and fodder? So quite a lot there, but hopefully that's something that you can get your teeth into. Who wants to start? Dennis, by the looks of things.
Okay. I would say, no, we cannot say it like this. Like, not all pesticides will affect all soil-dwelling organisms. It depends on the mode of action of the pesticide. So probably most pesticides are not just affecting the target species, but not all pesticides affect all soil-dwelling organisms, and probably no pesticides are affecting all soil-dwelling organisms. Yeah.
Okay. Yeah. Vera, do you want to join in?
I fully agree. Having to add, yeah, I guess indeed, like we need to be aware that indeed, like pesticides are by default harmful substances, but they are harmful for an intended past. So that is why we developed and applied them. And here, indeed, like the characteristics of the pesticide needs to be accounted when we are making assessments on the impacts on other non-target species. So that's exactly what Dennis said, that we cannot assume that all pesticides harm everything else. No, that's not correct. We need indeed to go look into the characteristics of the pesticides that are in the soil.
Lovely. Okay, I think that's really helpful. This is a follow-on question that relates to the question above from Hazel. And it'd probably be good if you explain some of these terminologies to the audience just to make sure that we're all in the same place. So where MEC exceeded PEC, has analysis been done to assess any impacts on soil fauna?
Not yet, I
Okay.
think. So there, as Vera told earlier, we sampled everything. So we sampled the soil samples also for microbial analysis. There have been earthworm samples be taken from all the case study sites and also of beneficial insects. But I am not aware if the insect paper is published yet. I know that the earthworm paper and the microbiome paper are not
Finalized yet, yeah.
Yeah.
Just to add, indeed, for make sure that everyone is on the same page, the MEC, it stands for Measured Environmental Concentration. That is what we measure in the labs, in the soils, in this case. And the PEC stands for Predicted Environmental Concentration. So that is a theoretical value derived from application records and theoretical degradation kinetics. So we notice indeed that rather often we had a mismatch between these two indicators. And here, I fully agree that when this situation of MEC exceeds the prediction, that further investigation is required here to address indeed like the reasons why it happens, but also the implications. And I guess in the paper of Danish, we reflect a bit on the possible reasons for it that can be related, for instance, with longer than expected persistence of the pesticides in soil. That is the most likely explanation for this situation. But indeed, a follow-up on the risk-specific site analysis makes all sense. And I'm sure that we will go there also once we integrate additional information from microbiome, diversity, and so on. Yeah, thank you for that.
Okay, no, that's great. And so people need to keep an eye on the papers coming out of Sprint, and you've clearly got plans in hand, so that's great. So let's just then think about this is a question from Leonard. So how were the case study sites and practices selected for assessments? What motivated you to select a diverse range of practices rather than focusing on specific production systems? Maybe this is more for Vera than Dennis.
Yes, well, that's a great question. Thank you. Well, there are definitely some projects that focus on specific crops, and when they do so, it's possible that you go deeper in terms of all the practices and so on. In our case, in the case of Sprint, we had this underlying foundation of holistic assessments. So we wanted it to understand the impacts of pesticides overall. And therefore, we decided also to look into all main crop systems. So how did these case study sites come together? Well, we started by identifying the main crop types in Europe. That was the starting point. And then we look for locations where this crop is of high relevance. For instance, for vineyards, we look into Bordeaux region. And then we follow up on the next step that was about recruitment. So it was a tiered approach. So identification of the crops, more interesting locations for that crop, and then also the logistical and the practicalities comes to it with the recruitment and interest of the participants. I must say that on this last step, also the public perception on the pesticide problematics also played a factor. So it was sometimes we had to invest quite a bit and be quite persistent on explaining the project and be out there to address any potential question or concern, so that we could have in the end the representativity that we were aiming at. And I'm very happy with the output, and that we can now make tailored recommendations overall. Yeah, I hope that answers the question.
No, that's great. Thank you very much, Vera. And I think that that's really important that we clarify that for people as to why these decisions are made, and we're just very open about what that means. Let's just go to the next question, which comes from Kimberly. What kind of alternative to conventional pesticides did you use in the case studies of the Sprint project?
What I'm doing. Can you repeat, Matt? I
Yeah,
missed
sure.
the first part.
So that's okay. What kind of alternative to conventional pesticides did you use in the case studies of the Sprint project?
So in each case study site, we focus on two management practices. So I mean, conventional production and organic production. So that is default in all case study sites. We did dig deeper into the practices conducted in each one of these fields. And this goes not only in terms of pesticide application, but overall land management. In the end, when we work towards this final stage of the project, and when we are putting together the transition plan, and when we identify opportunities and lock-ins and barriers, then we keep it very open to the farmers as well. And again, as I stressed right in the beginning of my presentation, we try to be also very engaged with all actors. So we have data, we have supporting data from these case study sites, and that is like the starting point, but it goes beyond that, and we acknowledge that. So the challenges and opportunities go beyond our 11 case study sites, goes beyond Europe and so on. So we try to reflect on the tailored specific situations, but try to upscale and put it in context as well, so that it's applicable, or at least should be accounted, considered for other locations and so on.
Lovely. Thank you very much, Vera. That's great. So then I'm hoping Dennis is going to return in a moment, because I just wanted to ask about figure 1. In regards to the paper, you state that 52% of pesticide residues were exceeding their detection limits.
Yes.
Have you quantified by how much each pesticide exceeded those limits?
Yeah, so we have for every measurement, we have a value. Like that's basically what was on. It's in figure 2 of the paper, I think. Or so we have the box plots. I quickly just can share my screen again, maybe. So this is basically here. So here we have for every single
Okay,
sample.
Just to hold you a moment, Dennis. I certainly can't see your screen.
You can't see my screen. Okay.
So just let's
try again. No.
There we go. It's arriving. I think there we are. There we go.
Yeah. So it's figure 3 of the paper. So we have here for every sample the actual concentration. So every sample that was over here the total concentrations, but in the supplementary data of the paper is a table with all concentrations for every single pesticide for every sample that exceeded the limit of detection.
Lovely, great stuff. Thanks, Dennis. And always a good lesson to all of us. And so there's much to be learned and much fun to be had in the supplementary data. There's always the appendices and the supplementary data are always a great place to have a little rummage around, isn't it? And just indulge your data questions and queries. Okay, another question from Leonard. Considering that 97% of the 201 soil samples were contaminated with pesticides, which locations were absent in their samples from pesticides? Was this because there were no pesticides in the, or the residue level was below what your diagnostics could detect?
So there were, it was 6 samples, I think, that were pesticide free, and that was one conventional sample from Slovenia, I think, and then the rest were organic samples from different countries. I think it was from Slovenia, one, 2 from Portugal, and 1 from the Netherlands. I can quickly look that up. So it's a conventional and organic field from Slovenia, 2 organic fields from Portugal, 1 organic field from Croatia, and 1 organic field from Denmark.
Okay. So, I mean, and I guess that this is the weight of Leonard's question, is that how confident can we be that they were actually pesticide free, or were they?
If they just we just not able to measure them.
Yeah.
So I guess like we were from the pesticides we measured, I think our detection limits are quite low, and we are quite positive that we measured them if they were there. But on the other hand, we just analyzed 192 pesticides. There are active substances or residues, and there are about 450 approved in the European Union. And we know that pesticides that were not analyzed by us have been applied. So I think rather that's the cause. If they would be contaminated, then because we didn't measure them, not because we measured and didn't find them.
Yeah. Okay. No, that's great. That's very helpful. And then also, let's just hold on and pause for a moment, that the rather interesting finding that we are asking about 6 sites that appeared to be pesticide free, whereas the vast majority of sites you were detecting pesticides, which is maybe something for us all to consider. So that's very helpful. So a question from Stella. How were the predicted concentrations estimated for both organic and conventional pesticides?
So we only predicted concentrations for conventional pesticides for 63 different fields. And we basically used the escape model. We programmed it in R to make it automaticize, but like it uses focused degradation kinetics and application rates, crop interception, and then different degradation kinetics and the DT50 values. And we kept like bulk density at standard 1.5. So relatively simple.
Yeah,
Okay.
but maybe if I may, I think it's important to stress that we kept it simple indeed, in line with also the procedure that is conducted at regulatory entities. But we adapted the application rates to the values that we retrieved from the interviews with the farmers. So we had all this information about application records. So we accounted for that in our predictions. So this comparison could be indeed like more realistic.
And our predictions, we also used the worst case field degradation times. So the longest that are used in the EFSA records from field.
Great, thank you. No, that's important, and I think also very helpful for people to understand again how these methods are doing. Very open, about how this is working. So next question I have is that what do you think explains the large discrepancy between the predicted concentrations and the actual concentrations? Do you think that the predictions have inaccuracies? Do you think that the existing pesticide residues or residue accumulations are not considered in predictions?
Like in our predictions, we could just predict from the growing season from 2021. So our first applications were in November or December 2020 for some crops, but as we do not have like a T0 sample to see if there is previous contamination, that can lead to inaccuracy, that we just have like legacy from years before. On the other hand, there can be just by spray drift from somewhere else. There are different sources. I guess that accumulation is, or like previous contamination is one driver of inaccuracy. Maybe the biggest, but we cannot prove that with just one sampling time.
Okay, no.
And
Vera,
if I
go on.
may add, because I think when we are talking about this MEC-PEC mismatch, there are here two different scenarios. Is
Sure.
when the measurement is above the predictions, and when the measurements are below the predictions. And when also the measurements are below the predictions, is also interesting to understand what can be the reason why this happens. And Dennis was talking here about legacy that justifies measurements higher than the predictions, but I think it's also important to look on the other dimension. And when measurements are below predictions, it also highlights potential transport of these pesticides from soil elsewhere. So it's not a straightforward. We do a comparison, of course, but there are here a lot of processes and that needs to be accounted. That may explain, at least partially, this mismatch.
Okay, no, that's really helpful. And I think, again, just helps explain what you've done and how you've done it. People can return to the paper. Now, I'm going to be a bit like a journalist interviewer now. And we've got probably one question coming up. We're going to exit on time. And so it's quite a long question. So it needs as brief an answer as you can manage. Okay, so this is from Ramos. Did you have a look at the influence of effectivity, which is 10,000 times the effect of DDT today, I think, of pesticides when looking into the distribution of pesticides, or only on the mass of the substances?
Only on the amount, only mass. So we did not calculate any factors in so far for especially like risk assessment. We were thinking about doing a risk assessment paper in that direction, but so far it's just mass.
Excellent and admirably succinct. Well done, Dennis. That's great. Because we are coming to a close, I'm afraid, friends. What I was going to say is we have lots of great questions. We will attempt to answer all of those questions and get back to people. We're not seeking to avoid tricky or difficult questions, or not allow people to make their contribution. We've just taken them in the order that they arrived. So thank you very much. We have captured all of the questions, and so we will get back to you. You'll see that there are multitudinous varieties of kind of resources that you can keep up with Sprint about. You've already heard that we have plans for further publications, so please keep your eyes out for those. Now, we did mention at the beginning that this is the first seminar of a series. So next time, please come and join us on the 27th of November at 12:30 to 1:30 Central European Time. If you're not on Central European Time, you'll just have to work that out for yourself. But it's pretty straightforward, isn't it? Most of the time. So I'd really, really like to thank Dennis and Vera for joining us today. That's really been, I appreciate all the work and effort. if you're that you've put in for today. I think next time Vera's joining us again with Aurea Hernandez. So, but I think we will look forward to that topic when we come to it. So we've come to the hour. Thank you very much again, Dennis and Vera. Thank you all of the backstage team who've been busy supporting us hidden away. So that's Honor, Jane and Charlotte who've been busy making sure all of this works very, very smoothly. So thank you all, particularly for your participation. Really appreciate the time that you've given us. We look forward to seeing you again. Please do keep in touch. You've got lots of ways of doing that. And, you know, if you have questions, we're really here to hear from you. So thank you very much, everybody, for your time. Thank you for your participation, and done in a really lovely way. So thank you again, and we'll call it closed.
Thank you.
Thank you. Thank you very much.
Okay, we've got one person who's still with us.
It might be one that joined and are not currently in front of the computer.
Yeah, that's okay. Okay, great.
Thank you very
everybody.
much.
Really good.
Thank you.
Yeah, just a quick question. We did have a question in the Q&A whether the slides would be circulated, and I wasn't quite sure what you wanted to do with that. So, and whether it's actually possible. Have we got all the?
Can do that. Yes.
Yeah, fine for me. I think also we can put it also accessible via the Sprint website. We can put there like the link to the record and also supplementary materials or something like that, maybe.
Yep.
Well, and then obviously there's some questions that we didn't get to. So
Yeah.
do
I think we can also like follow up either by email directly if we have the emails of these people.
We have. I think for most we have.
Okay.
Or again, we can always like make even like I guess one session at the very end that if there are any unleft answers to be done, so we can also make at some point in the webinar also like around the table overall on the works and so on. So maybe. Yeah.
Great. Okay, lovely.
Thank you so much.
Thank you.
Thank you.
Thank you. That was great. Well done, Honor. Well done, Jane.
Thank you. Thank you,
Thank you.
everyone.
All right. Okay.
Bye bye.
See you.
Bye bye.
Bye-bye.